All right, I'd like to welcome everyone to the last Spring 21 Polyploidy Webinar today. And today we have two great talks. Up first is Dr. Brittany Sutherland. Brittany, as many of you know, was a postdoc in my lab for a couple of years as an NIH postdoc, and is currently the program and research scientist for the Consortium for Plant Invasion Genomics at the University of Louisiana at Lafayette, otherwise known as CPING. Brittany grew up in Kentucky and earned her undergrad from Western Kentucky University, and then completed a master's at Morehead State University in teaching. And for maybe that don't know, Brittany did a bit of a stint as a middle school and high school science teacher, and is just one of these just great presence in a classroom, and but found that it wasn't exactly for her. Went back to college and went to pursue a PhD at the University of Virginia. Where she pursued really what I think is really cutting edge and interesting work on the reproductive isolation of polyploid species and reproductive isolation within polyploid species complexes. And today, the work she's going to tell us a little bit about today is along those lines, with this, I think, wonderfully inspired title, Where the Rules Are Made Up and the Dosage Doesn't Matter, Potential Conduits for Interploidy Gene Flow. And with that, I'll let Brittany take it away.
Awesome. Thank you very much, Mike. And thank you all for coming to our last session of the semester before summer. And Mike gave a lovely sort of rundown of what I've been doing, and I am basically circling back around to my PhD work for this talk for the most part, and talking about some of the interesting things that can happen in polyploid complexes and the problems we currently have with studying them and what we should maybe look to doing in the future. The good thing with a talk like this is that I'm talking to a bunch of polyploid nerds, so I don't have to spend the first couple of slides telling everybody why polyploids are great and interesting and we should all love them. So I'm just gonna sort of jump in sort of in the middle, and if I can get my computer to work. So we all basically know that polyploidy is evolutionarily important. We know that it seems to be associated historically with sort of these key innovations, the origin of land plants, the origin of seed plants. It seems to be correlated with major ecological changes. There's a lot of polyploidy that seems to be associated with the KT boundary. There's a ton of morphological and physiological changes that can happen after polyploidization that can cause changes in habitat and pollinators, and it mediates lineage divergence and speciation. And that last point is really going to be the focus of my talk. How does a change in ploidy change species or not change species? So for a long time, we sort of had this idea that a change in ploidy was basically instant speciation, because we knew that when you went from being diploid to being tetraploid, a lot of sort of reproductive barriers could appear, both prezygotically and postzygotically. We know prezygotic barriers, again, when you change ploidy, you often get pretty substantial morphological changes, although not always. And those morphological changes can change, habitat preferences and definitely pollinator syndromes. You can also change phenology, as we see in Justin Ramsey's Achillea work. Just changing the genome copy number changes the flowering time. We also have a lot of postzygotic barriers. So we can have hybrid inviability, where if you cross a diploid with a tetraploid in either direction, as we have here with, I believe this is brassica, you don't get the right amount of endosperm development. And so you get sort of a breakdown of seed development, and either, they don't germinate or they're sort of weak and don't make it to adulthood. If you make it to adulthood, you can often be infertile if you're sort of a triploid hybrid. And for humans, we often use this to our advantage agriculturally. So those of you all who like seedless watermelon, you can thank triploid hybrid watermelon for that. So there's all of these sort of large scale things that can happen when you change your genome that seem to support this idea that you're essentially a new species or a new isolated lineage once you've duplicated your genome. So now that I've completely set up that straw man, we find lots of examples of cases where this doesn't seem to play out. So when you look in Capsella, you can still see gene flow between diploid Capsella rubella and tetraploid Capsella bursipastoris. We see, multiple recurrent originations of these mimulus hybrids in the UK. And in perhaps what might be the best studied sort of autopolyploid system in Senecio carniolicus, we find that when you have tetraploid and hexaploid cytotypes growing in close contact, like we have here in panel C, it's kind of hard to see on this screen, but if you see all of these little purple dots in the middle here, those are pentaploid hybrids that have formed between the tetraploid and hexaploid cytotypes. So, there's been this longstanding idea that you have this reproductive isolation, but there's exceptions. And that sort of was the point where my work began on Campanula rotundifolia as a grad student. And this is a pretty good system for studying reproductive isolation between cytotypes because there's 3 dominant cytotypes. So it's not too big a polyploid complex, but it's not just diploids and tetraploids. And importantly for my work, it is outcrossing. A lot of polyploids, when they duplicate their genome, become selfers. And so first I wanted to just see, do the different cytotypes cross with each other? And when they do, what kind of offspring do you get? So I started out in rotundifolia, there's largely tetraploid populations with sort of pockets of diploids and hexaploids. So I crossed, 2 test diploids and test hexaploids against a slew of sort of tetraploid populations. And I did find that there were differences in reproductive isolation. So, the composite sort of reproductive isolation index, which was basically, the product of seed number, germination rate, and fruit number, we do find that while both of them have pretty high reproductive barriers, there's definitely a difference. So the, offspring produced by tetraploids and hexaploids in either direction did a little bit better and survived a little bit better than the ones produced by diploids and tetraploids. And when we look at germination, there's a huge difference. So when you cross diploids with tetraploids, very few of those seeds germinate, about 10%. Whereas when you cross tetraploids and hexaploids, you get 60 to 70% the germination that you get in sort of our controlled homoploid crosses. So this led to, my work and other work in the field has led to sort of this updated idea that while the instant reproductive isolation idea works pretty well for diploids and tetraploids, oftentimes among higher ploidy levels, these barriers appear to be weaker. However, the long-term, consequences of those weaker barriers are still pretty unclear. And so what I'm going to talk about next in this talk is there's, sorry, train of thought completely left. As a follow-up to that first study where I was crossing diploids and tetraploids and hexaploids, I then, based on those results, decided to do a follow-up. And this was not a follow-up that I had originally planned to do. So it was quite small, and I would love to sort of, do another version of this at some point. But, we find that interploidy hybrids, they're there. We know that pentaploids exist, and we seem to find more pentaploids than we do triploids. But then what happens with these pentaploids? Are they basically just a sink where the tetraploids and hexaploids are throwing gametes into this and wasting resources and not getting anything back from it? Or can those pentaploid hybrids actually allow for some gene flow and, they're sort of selected to be maintained at least to sort of a low degree? So I, with those questions, I did some follow-up work after that first study by taking some of the better performing populations from those early crosses and doing back crosses to the parental cytotypes. So this is very much sort of a stacked deck, and I want to be honest about that up front. So I took sort of the populations that gave us the most triploids and the populations that gave us the most pentaploids, although among pentaploids, we had a sort of a range to choose from. Lots of our populations of crosses there gave us pretty good germination, lots of individuals to choose from. But with triploids, I just sort of have to pick and choose. So we wanted to see, do they back cross to the parental cytotypes? So can these hybrids potentially serve as a conduit for gene flow? And do we see any evidence of any kind of gene flow in the field? So we know there are naturally occurring mixed ploidy populations. So, if there is ongoing gene flow between tetraploids and hexaploids, as opposed to diploids and tetraploids, we should be able to see that. So again, so those are my questions. So to do the back crosses, again, we had these 3X's and 5X's that were, chosen specifically because they sort of had the best chance of survival. We back crossed the triploids to diploids and tetraploids, and the pentaploids to tetraploids and hexaploids. And we use the same parental populations that created these in the first place. So we didn't want to, you have genetic distance conflating anything. And so what we find is that, sorry, you guys are blocking part of the screen that I need to see. There we go. So what we find is basically there was almost no offspring created between triploids and their parental cytotypes. We had a very small number of seed set and a little bit of germination in some of the individuals, but essentially nothing survived to adulthood. Whereas our pentaploids seem to do pretty well. On average, we had about 70% seed set, about 80% of those seeds germinated. And, so composite fitness wise, we had quite a number of successful back crosses. And interestingly enough, there was a directionality to it. The pentaploids seem to do better when you cross them against hexaploids than when you cross them against tetraploids. So, with the back crosses, the pentaploids definitely could serve as a conduit for gene flow. The triploids definitely can't. We just basically don't have any. So next we wanted to see, do we find any evidence in the field of these patterns? So again, I said there's 4 known contact zones that I have. I know that there are other people working in this who have lots more, and hopefully in the fall they'll be interested in doing a talk. But of those 4, we had 2 that were diploid tetraploid, 2 that were tetraploid hexaploid. So I went through and basically sampled leaf tissue from every plant that I could that was at least 1 meter apart. Estimated genome content using flow cytometry and genotyped these at 16 microsatellite loci. So what we find from the flow cytometry is basically what we would expect for diploids and tetraploids, in the sense that we had a nice sort of cluster that grouped at sort of what we expect the diploid size to be in both of our contact populations, and then a nice cluster of tetraploids. By contrast, in the tetraploid hexaploid populations, we find kind of a smear. So we find a little bit of a clustering of tetraploids that seem to be kind of off on its own, some hexaploids that fit, the known size, and then some individuals that appear to be pentaploid. And then we find some that are sort of 5.5 and 4.5. So based on this, it seems like there's still ongoing reproduction, at least in the 4s and 6s. And so next we wanted to see if there's any, genotypic evidence here. And we find, I can talk about this little guy afterwards if we have time, but essentially we find sort of clean groupings between our diploid individuals and our tetraploid individuals in both of our populations from a principal coordinates analysis. Again, to contrast, here's our 4X and 6X populations. There are no clean groupings of any of our cytotypes, and the pentaploid and putative aneuploids are just sort of mixed in there with everybody. The only signal we maybe found was a little bit here with some tetraploids that were on a cliff sort of separated from all of the other plants, but we found nothing cytotype based. So again, pentaploids could maybe serve as a conduit for gene flow. Triploids, I think we pretty much put that to rest. And we do find evidence of gene flow occurring in tetraploid hexaploid populations. So they do have higher fitness, but do they sort of ongoing serve as a conduit for gene flow? And while the evidence that I have points to that, I wouldn't say that it's conclusive. We've gotten one step further. We know that these hybrids are in some cases fertile and can back cross to the parentals, but aneuploidy seems like something that plants aren't usually happy with. So these could still be a sink ultimately, just one step removed. Or it's possible that we could be seeing a conduit for ongoing gene flow between the stable cytotypes. So this is the question that I haven't answered yet. And it's something that I would like to sort of just pitch to the group broadly. There seems to be more aneuploids showing up in a lot of people's papers these days. So we find it in my work. We found it in other work in rotundifolia. There was a paper that came out last year in Julia Wilson's lab that basically cytotyped everything she could find in the UK, and does have these individuals that sort of fall in between what we expect to be sort of good tetraploids and hexaploids. There was a paper that came out just a few weeks ago out of, I think, the Holver lab in Austria, looking at Senecio carniolicus, which is, again, one of sort of the best studied systems here. And so what you're seeing here in this graph, we have relative genome size of pollen versus the pollen donor. So you would expect, your gamete to be roughly half the genome content of, the somatic cells. And in the pentaploids, it sort of runs the gamut. So some are considerably larger than half, some are considerably smaller. So we're getting sort of this range of chromosomes that are being passed on to these gametes. But when you look at offspring made from these crosses, the pentaploid crosses do really poorly. But a lot of the tetraploid pentaploid and hexaploid pentaploid crosses, maybe don't have great seed viability, but the ones that do show up survive quite well, and their sizes are comparable to homoploid crosses. So, we're getting more aneuploids here, and aneuploidy is traditionally not thought of something that should stick around very long. Genome dosage is something that's tightly controlled in a lot of systems. So why are we finding these aneuploids? First, we do know that plants tolerate it more than animals do. In crop systems, there's a lot of monosomic and trisomic plant lines for assessing gene dosage of particular chromosomes. And we know that some chromosomes are tolerant of different dosages than others. So it's possible that these aneuploids that are persisting in Campanula and Senecio and these other systems, that they could be specific chromosomes that are being overrepresented or underrepresented. And so that could cause sort of, differences in selection that could happen with gene flow. But there could be other things going on with these. So long-term, we could be having a case where we're just catching things at a weird moment in time and we're just taking snapshots. So this here is showing kind of a misleading snapshot of, this giant moon. And it's possible that these populations that I've been looking in Campanula are only 50 or 100 years old. And if we wait another 50 or 100 years, one cytotype will have eventually dominated. So we could just be catching these at the wrong time. They could divide niche space and, find, little microhabitats where tetraploids do well and hexaploids do well. And we find this rate of pentaploid and aneuploid formation decrease, or they undergo reinforcement again to do the same thing because they're tired of sort of wasting their gametes. Or we could have some degree of hybrid swarm that persists in the long term, where some low degree of aneuploidy is around and they're semi-fertile and they're still contributing to the population. But we don't know yet. So one of the things that we need to do, I think, as a community, is we need to do more study of natural autopolyploids. A lot of the talks that have been in this series that have been great are allopolyploid focused. And I'd love to see more autopolyploid work just generally. But in some cases it's harder, it's harder to find them in the field. Oftentimes, new neopolyploids are cryptic, they may not have substantial differences. But one of the things that I think I would love to see as a community is, can we get better autopolyploid model systems? Right now we seem to have some that we have a lot of genomic data for, especially in sort of the ag circles, and then some that we have a lot of ecological and cytological data for, and we need those to come together. So that, I think, will help move the field forward. There's a lot we can learn from ag. A lot of the autopolyploid systems that have the best genomic data are crop species, things like potato. And I think that's a place that we could learn from to then look at natural populations. We also need to, all of the people who are early career folks, learn how to count chromosomes and do more of it. So, flow cytometry is a great tool and counting chromosomes is a great tool. They're not sufficient on their own. Both of them need to be done. So we need to be able to see how many chromosomes we have, and then use that information to build genomically and see, okay, if we have, say, an individual that has 3 extra chromosomes relative to its parents, which chromosomes are those? So, that's where the genomic tools are going to come into play. So all of these kind of need to come together. So we know that autopolyploid complexes are likely undercounted, just because they're harder to find. And it seems like the more we look for them, the more polyploids we find in certain populations where we maybe didn't expect them. We need more fine-scale sampling of autopolyploids in these contact populations with multiple data types, chromosome counting, FCM, genomic data. And we need to probably reevaluate our assumptions on odd ploidy individuals and aneuploid individuals that we find. They may not be just, dead ends, and they might be something we need to follow up on. But as with everything else, we need more data. That's sort of the perennial gripe of everybody, right? So ultimately, we need more data as a group, and hopefully in the fall and going forward with this series, we can come back and talk more after we've learned some things. So with that, I'd like to thank my PhD lab, where I did a lot of the work that I showed you here, as well as Mike for being a very patient and supportive mentor while I was in Arizona, my current lab with Nick Coyers at Louisiana. And with that, I hope everybody has a good summer. And I'm happy to take questions.
All right, well, thank you, great talk. As always, folks can go ahead and pop their question in there, or I see Camille's got his hand up already. Camille, go ahead and pop on your audio, video, and ask away.
Yeah, great talk. I'm quite fascinated by this barrier between the higher ploidy levels and how the aneuploids still can produce viable gametes, because that's something animals don't really like to do. So I wonder, like, how do the gametes look like? So you imagine that there are just like some chromosomes missing, or if a pentaploid produces a gamete, it's either 4, 6, 5, like.
So I have not done any flow cytometry with gametes myself. I can tell you what they look like in the microscope. At least in rotundifolia, they are incredibly variable when you see those gametes from pentaploids. With tetraploids and hexaploids, you get these nice consistent diameters, except for, the low level of unreduced gamete formation. So you'll have like all of these that look the same, and then like a beach ball. With the pentaploids, they run the gamut. They all stain as though they are viable, but I know with that test, it's pretty conservative and can overestimate the number that are actually viable realistically. But we did find that these individuals were creating back crosses. So I don't know what chromosomes are involved there. That follow-up study was basically the last 4 months or so of my PhD, and then I moved halfway across the country. So at some point I would love to come back and, come back to this system. I've got plants growing right now that I want to do some FCM on over the summer. But I would love to talk to the people who did that paper, the one that just came out, Pascola and that group, because it looks like, at least from their data, that their chromosomes in the pollen were all over the place. And I want to know if that's what they actually found. So
yeah, because I had an associated thought that if for any reason there is a higher chance for the pentaploid to make a triploid over diploid gamete, then it would also explain why it's easier to form a hybrid with a hexaploid over a tetraploid. So yeah. I was just wondering if there can be any of those like meiotic drive mechanisms involved in this. Well, but that's probably completely.
I mean, there probably are. You know, I don't have a lot of experience with that, but you know, with so much of this, as someone just said in the chat, you know, these are hard populations to find, and so we just haven't done a lot of the work, or the work is 70 years old. So I think Carl's got his hand up.
Yeah, I think Carl, Jonathan actually posted his request there first, and Carl's put his hands up. So maybe Jonathan, then Carl. How's that?
Oh, okay. I'm happy to defer to Carl. Carl probably has something more important to say.
Untrue. Go ahead, Jonathan.
I'll let you guys argue about it.
Well, I'm just sorry. I'm really sorry, Brittany, that speaking of 70 years ago, I'm sorry that Stebbins wasn't here to listen to your talk, or Vern Grant, and you know, I'm thinking of the classic sort of agamic complexes that you know, all of us learn when we're about 4 years old, and we grow up knowing about them. And it seems to me that plants are pretty respectful of their complement of chromosomes and its purity when they're diploid, and that as ploidy level increases, they become decreasingly respectful of complements of chromosomes, more likely to interbreed with anything under the sun, more likely to spin off aneuploids. And all of that is a great big mystery when you think about how all of these chromosomes are packed into the nucleus and how they manage to do everything that governs growth and development and reproduction. So it's really kind of a bizarre observation in nature that I don't think we have any explanation for at any kind of fundamental biological level, and especially when you consider how aneuploidy is so bad in terms, like Jim Birchler's studies. He wrote this beautiful poem about how aneuploidy is bad. I don't know if you've seen it.
I've heard of it. I haven't seen it.
Yeah. But what do you make of this sort of increasing propensity to be badly behaved as chromosome number increases?
I mean, that's an excellent question. So a couple of things, maybe. I don't have a good sense from reading the literature how many chromosome counts were done from individuals that were just plucked in nature versus ones that were, you know, at some point plucked in nature and then cultivated in a glass house somewhere. So I'm wondering if some of these, you know, weird aneuploids that we find now by looking at leaf tissue with flow cytometry just didn't persist in the greenhouse, and so they were sort of selected out. That's a wishy-washy answer, but you know, in terms of like agamic complexes, I think you're absolutely right. I mean, the first system that I cut my teeth on as an undergrad was rubus, and rubus ursinus specifically, which has like 5 or 6 different cytotypes in California. And, you know, one thing that I think would be fascinating would be to look at, you know, what's the actual frequency of different mating systems in polyploids, because a lot of these that have the really bizarre chromosome numbers, you know, they don't necessarily go through sexual reproduction. And so they might be able to sort of get around some of these barriers that appear during meiosis. But that doesn't explain everything. You know, rotundifolia is perfectly sexual, and so are lots of other polyploids. I think that we need sort of another round of study. I know in a lot of cases, some of these chromosome counts that people have looked at, when they go back and look at them again, they don't necessarily find the same numbers that people found before. I don't know. Yeah, there's a lot here, and I don't really have good answers for these yet. Sorry about that. So talk to me in 5 or 10 years, and hopefully we'll have some things to say. Man, I would love to figure out Claytonia. I actually, while I was talking to Mike about, starting as a postdoc, that was one of the systems that I looked at as, because there's these, kind of cool polyploid series where you've got, I guess, Virginica and Rubra and some other things where like all of them have this sort of step ladder of 2, 4, 6, and sometimes 8. The problem is a lot of these are really hard to study. I mean, I think doing Claytonia in the lab would be a nightmare. There's a reason I'm not working on Rubus or Sinus right now. I mean, it's much harder to convince to be annual than Campanula is, not to mention it bites. So, with things like that, with like Ursinus, the chromosome, the flow cytometry for those, for the most part, was from about 20 years ago. And I don't know how well you can really differentiate between a 10x and 11x and a 12x. So some of these I think we should probably revisit and revisit genomically. But, I don't want to say that all of the old data is wrong, because I'm sure a lot of it's great. So.
All right. Well, I think if we're getting into Mischa's time here. Carl, are you okay waiting until the very end? Because we always have plenty of time at the very end. All right, cool. I get the thumbs up from Carl. All right, well, we'll go ahead and thank you, Brittany. And if there are any other questions, please save those until the end, and we'll go ahead and let Mischa share her screen now.
Sure, let me get set up here. And while I'm getting set up, I just want to say I'm so excited that by happenstance, Brittany and I are on the same week, because I agree with so many of the conclusions that she stated, and I think our talks fit very well together. So this will be super exciting.
All right. Well, I am as well, and I appreciate, Mischa, you're the last talk of the spring semester, but certainly not least. And so Mischa Olson is our second speaker. Mischa was first introduced to the wonders of polyploidy as an undergraduate researcher in Jonathan Wendell's lab at Iowa State. Mischa is now a postdoc in the lab of Wojtek Pawlowski at Cornell University, where she recently defended her PhD. And she tells me here that she's currently looking for jobs in London. So this is an opportunity for those of you in the UK to actually hire Mischa, who does really outstanding work. And today she's going to tell us about some of her observations and work on meiosis in neotetraploid maize. And I'm really, really excited to hear about this. And so with that, I'll let Mischa take it away.
Great, thanks. I'm super excited to share sort of 3 little vignettes from my thesis work today, and hopefully bridge the gap between some of these ecological studies and things that Brittany was talking about. And also, I think there's a huge expansion in the meiosis field in the past couple of years in terms of our understanding of these chromosome dynamics during meiosis. And I think as a polyploidy field, we'd really benefit from zooming in a little bit on meiosis. So I'll try to do that a little bit today. So again, like with Brittany, I don't need to go into too much detail about how polyploid plants are agriculturally significant and inherently biologically interesting in lots of ways. But it's always fun to see the pictures and remember just how cool they are. In addition to plants currently retaining multiple sets of chromosomes, we know that throughout the evolutionary history of eukaryotes, there have been all of these ancient polyploidization events, and that this phenomenon is particularly prevalent in plants. And this has led to the hypothesis that polyploidization can provide the foundation for the creation of new variation as these ancient events tend to occur before the radiation of very speciose and diverse lineages. But it's complicated, like we were just talking about. Newly formed polyploids, also called neopolyploids, can be smaller than their diploid progenitors. Here you can see 2 diploid maize plants on the left and then 2 tetraploid maize plants. Like Brittany mentioned, neopolyploids can take longer to flower and they can have low fertility rates. And these can be indicators of genomic instability. In fact, when we look at the karyotypes of new or young polyploids, like these young tetraploid trigopogons, we can also often see extensive aneuploidy and chromosome fusion. And this genomic instability often proves fatal. Some studies have shown that the majority of neopolyploids go extinct. So one of the big questions we all kind of wrestle with is, how can polyploidy have such a profound impact on the tree of life if most of them go extinct? Well, after polyploidization comes diploidization. And that's the process by which over evolutionary time, a polyploid can revert to a diploid-like state. So here just is a cartoon. We have an example polyploid with 2 parental chromosome sets A and B. As soon as the first few generations after polyploidization, there can be gene loss. And a polyploid can lose more genes from one parental genome than the other. And this is called biased fractionation. One or both genomes can lose even more DNA, and there can be chromosome rearrangements and fusions, like we saw in Trigopogon. And then over the course of evolutionary time, this A plus B genome plant can undergo another polyploidization event, and this will restart the cycle. And really, it all comes down to meiosis, at least from my perspective. Many of these processes of diploidization, like these chromosomal rearrangements and large amounts of DNA loss, occur via the meiotic recombination pathway. Many of the other ramifications of polyploidization, like chromatin remodeling and accessibility changes, changes in DNA methylation, these are known to impact these cytological processes and also modulate recombination patterns in their own right. But the connections between these processes haven't really been systematically studied. So the incredibly and perhaps overly ambitious goal of my dissertation was to dissect this loop and understand the direct impact of polyploidy on meiotic molecular mechanisms. So the lengthiest stage of meiosis, and where a lot of the action happens, is prophase 1. And we divide it into 5 substages: leptotene, zygotene, pachytene, diplotene, and diakinesis. Sorry for all the terms. During prophase 1, there are 2 essential and inherently linked processes which occur. The first is recombination, which we're all familiar with as the exchange of genetic material between 2 chromosomes, which ensures proper chromosomal segregation and also has the added benefit of generating genetic variation. The second process is pairing, which is defined as the close association of 2 chromosomes during zygotene and pachytene. A related but distinct process from pairing is synapsis. And synapsis also occurs during zygotene and pachytene and involves the installation between 2 chromosomes of a zipper-like structure, which is formed by the protein ZIP1. Recombination, on the other hand, is initiated much earlier in prophase 1 during leptotene. A topoisomerase-like protein, SPO11, initiates recombination by making double-strand breaks, or DSBs, in the DNA. And these double-strand breaks are resected, which results in the overhangs of single-stranded DNA, like you can see on the bottom. During zygotene, 2 proteins, DMC1 and RAD51, associate with this single-stranded overhang, and they facilitate a process called single-end invasion. And during single-end invasion, this overhanging piece of DNA invades a double-stranded piece of DNA. And this is a really key process because this invasion of the overhang allows for a homology search to take place. And due to the association of Rad51 with these single-stranded overhangs, we can use it as a marker for double-strand breaks, which you see here. And after these single-end invasions, now these are all recombination intermediates, and they're funneled into a variety of pathways for resolution. In mid to late prophase 1, there are 3 ways that these intermediates are resolved. First, they can be resolved as non-crossovers, which is the most simple. The double-strand break is simply patched by DNA synthesis. You can see that in the center. The 2 other resolution pathways involve crossovers, and breaks can be resolved as class 1 crossovers, which are subject to interference, or class 2 crossovers, which are not subject to interference. In most diploid lines of maize, there are 400 to 500 double-strand breaks formed per meiotic cell, but only 20 of these are repaired as crossovers. And which breaks are repaired as crossovers and where is a huge active area of research and something that the other people in my lab work on a lot. In the latest stages of prophase 1, you can easily visualize chiasmata, and these are the physical manifestations of crossovers. And importantly, the chiasmata are the outcome of both of these processes. First, recombination, it indicates that a double-strand break has been successfully repaired as a crossover. And second, a pairing interaction was also successful. When chiasmata link to homologous chromosomes, like in this example, it's called a bivalent. So chromosomal interactions during diploid meiosis follow that outline. And we know that most diploid plants make bivalents at meiosis. Studies of meiosis in polyploids have previously focused on crossovers and the outcomes of meiosis. In new polyploids, there's a tendency for more than 2 chromosomes to be linked, forming multivalents. And some polyploids have evolved stabilization mechanisms to reduce multivalent formation, like reinforcing homologous pairing or reducing the crossover number to only 1 per bivalent. And by doing that, you effectively eliminate the possibility of multiple chromosome connections. But all crossovers are the outcome of these recombination and pairing mechanisms. But we rarely look earlier in meiosis to understand how the dynamics of these mechanisms are impacted. So that's my question. What is the direct impact on these mechanisms? So to begin to answer this question, I looked at meiosis in maize, which is a fantastic plant model for studying meiosis. And Jim Birchler conveniently has made tons of different maize tetraploid and aneuploid and all sorts of different maize lines for us to play with. I focused on 3 neotetraploids and their diploid progenitors. So first, there are 2 homozygous autotetraploid lines, and they each have 4 identical chromosome sets. The third neotetraploid is something I call a heterozygous autopolyploid, in that it has 2 distinct parental genotypes. Now, the definitions of the term autopolyploid and allopolyploid, we all discuss vigorously, very regularly. And often we strictly categorize into types when a spectrum may be more appropriate. For example, here, this heterozygous autotetraploid has 2 different parental genotypes, or 2 different parental genomes, and they're from within the same species. Its meiotic behavior, which we'll see in a little bit, has some similarities to other autopolyploids. But these 2 inbred lines of maize, Ohio 43 and B73, have more sequence diversity between them than do humans and chimpanzees. So in some respects, it's allopolyploid-like. So today I'm going to tell you about 3 little vignettes from my thesis that I worked on to characterize the meiosis in these lines. First, I wanted to see if recombination mechanisms are impacted, specifically in early and mid prophase 1. Second, I wanted to understand if meiotic gene expression is significantly altered. And finally, I wanted to see how polyploidization impacts the nuclear and chromosomal dynamics in prophase 1. So to address the first question, I took primarily a cytological approach. I was able to embed myocytes, which are cells undergoing meiosis, in a polyacrylamide pad, and this preserves their 3D structure, and we can image them in 3 dimensions. So here we're scrolling up and down through the Z-axis of the cell. And this is in zygotene, which is mid-prophase 1. DAPI is a fluorescent dye which stains AT-rich regions of DNA. That's in red. In green, you can see Rad51, which, if you remember, binds those single-stranded overhangs and marks double-strand breaks. In blue, we have ZIP1, which is the central element of the synaptonemal complex. So in this cell, which happens to be tetraploid, you can see numerous double-strand breaks and that the chromosomes are starting to pair and synapse. Using this technique, I compared the number of double-strand breaks between different diploid and tetraploid lines at different stages of prophase 1. Here you have zygotene on the top and pachytene on the bottom. And these images are just slices through a 3D nucleus, but you can already see that there's a large increase in the number of double-strand breaks in the polyploid lines at both stages. So in quantifying these data, here we have early to mid zygotene, which is when the peak number of double-strand breaks occurs in the cell. In this plot, each dot represents a cell, and each genotype on the x-axis has measurements from at least 2 biological replicates. In the 2 diploid inbred lines, all the way on the left, we see an average of 400 to 500 double-strand breaks. And importantly, we also looked at the diploid F1 hybrid cross, and we see a similar average. In the 2 homozygous autopolyploid lines, we see an increase in, but not a doubling, of the peak number of double-strand breaks. But in the heterozygous tetraploid line, we do see double the number of double-strand breaks, as in the diploid lines. Now, why the double-strand break numbers are elevated in this pattern is not 100% clear. There's a hypothesis in the meiosis field that because the single-stranded overhangs resulting from double-strand breaks function in a homology search process, that the production of double-strand breaks is linked to the progression of pairing in a feedback loop. This hypothesis could explain these data, in that the homozygous tetraploids have 4 copies of every chromosome, so perhaps it's easier to locate a pairing partner and fewer double-strand breaks are produced. In the heterozygous tetraploid, there are 2 distinct chromosome sets, so perhaps more double-strand breaks are produced to aid in the location of the preferred pairing partner. However, no evidence for this feedback loop currently exists in maize. A lot of work has been done in yeast, but yeast and maize are very different. And at this early stage of prophase 1, it's not clear that enough time has elapsed for a hypothetical feedback loop to have taken effect. If we look later in prophase 1 in pachytene, in diploids, we see that the vast majority of double-strand breaks have been repaired, and only 20 to 30 remain. In the polyploids, we see a persistence of double-strand breaks, and this indicates that either new double-strand breaks are being made or that double-strand breaks are taking longer to be repaired. And we can't distinguish between the two because these are fixed cells. So this is not the same cell in pachytene that I imaged in zygotene. Like Brittany was talking about, it's a snapshot. Interestingly, this persistence seems to be less severe in the heterozygous autotetraploid line. In maize diploid mutants, which are defective in proteins involved in pairing, we also see this persistence of Rad51 foci. So this persistence indicates that there are problems with pairing in these neotetraploid lines. Because we see these disrupted double-strand break patterns and a potential perturbation of the homology search process, I evaluated the progression of synapsis by tracing the synaptonemal complex in 3 dimensions in at least 10 cells per line. In each of the 3 diploid lines, I was able to trace on average 10 synaptonemal complex lengths, and this represents the 20 paired and synapsed chromosomes of diploid maize. In all 3 neotetraploid lines, however, I did not encounter a single cell where I could trace only 20 synaptonemal complex lengths, which would represent 40 paired and synapsed chromosomes. In fact, as you can see in the table, the average number of SC segments traced per cell was significantly higher, indicating that synapsis never completes in these cells. Additionally, in all 3 neotetraploid lines, I observed partner switches, which indicates that these issues with chromosome pairing have persisted. And these synaptic partner switches are often a prelude to the formation of tetravalents or multivalents. In working to quantify recombination outcomes, we counted the number of bivalents per cell in all of the tetraploid lines. And we see that the number of bivalents in the heterozygous line is higher, which indicates that it has more of this allopolyploid-like meiotic behavior. However, in all 3 neotetraploid lines, including the heterozygous line, in diakinesis spreads, I see formation of multivalents and large clumps of chromosomes which don't spread, and they're highly interconnected. And this is indicating potential associations between non-homologous and even non-homologous chromosomes. In metaphase 1 and also anaphase 1, I see lagging and misdirected chromosomes. So these disruptions are really having an impact. Because it's really difficult to quantify the number of bivalents and chiasmata in these cells because of the clumps, I measured the overall success of meiosis by the ability of the plant to produce viable pollen. And so on the top, we have the diploid lines, and we see that the pollen viability on average is 80 to 90%, which is consistent with inbred lines grown in growth chambers. In the homozygous tetraploid lines, which are on the left and the bottom, we see a large amount of plant-to-plant variability. So the viability levels range from 0% to 60%. And in the heterozygous tetraploid, the viability levels are generally much higher, though on average, they're still lower than the diploid lines. So if we return to our question, we find that yes, the double-strand break dynamics are altered early in prophase 1, and we see a disruption of the homology search process, which persists to impact fertility. So then I wanted to understand if these patterns and disruptions are due to the misregulation of meiotic genes. Sorry, just gonna take a quick drink. So in comparing gene expression across samples using RNA-seq, the common mode of analysis normalizes expression based on transcript abundance in a sample. But this essentially compares the concentration of each gene transcript between the samples, assuming the samples have the same total transcriptome size. But work from Jeff Doyle and Jeremy Coate and others has shown that the transcriptome size is often impacted by polyploidization. So in order to accurately assess the gene expression, we co-extracted RNA and DNA from seedling leaf tissue and also from isolated zygotene anthers. So here you can see the total transcriptome size per cell, which is total RNA, including rRNA and non-coding transcripts, etc. In the leaf tissue on the top, you can see that the transcriptomes of the tetraploids are about double those of the diploids. And in the anther tissue on the bottom, in B73, we see a doubling, but a less than twofold increase in Ohio 43 and the heterozygous tetraploid. If we look specifically at the mRNA transcriptome size, we can see that in the leaves on the top, the Ohio 43 and heterozygous tetraploids have a 4-fold increase compared to their diploid progenitors. However, in the zygotene anthers on the bottom, the mRNA transcriptome size mirrors that of the total transcriptome. So we see some really interesting tissue-specific effects. Using these transcriptome sizes, we can quantify gene expression levels per cell by calculating the expression level per amount of DNA and then multiplying by the amount of DNA in the cells. We then ask which genes are differentially expressed in all 3 cross-ploidy comparisons. So for example, in this leaf Venn diagram, if you look at the upper left-hand circle, those 200 genes are differentially expressed 2-fold or more in comparing the B73 diploid and tetraploid, and they're not shared with any other cross-ploidy comparison. However, if we look at the overlap region between Ohio 43 and Ohio 43 by B73, we see over 16,000 shared differentially expressed genes. And this mirrors the large increase in the mRNA transcriptome size that we saw. There's a lot to explore in these data, and if we look specifically at the anther tissue, we see a really low number of shared differentially expressed genes. We look to see if any Go categories were significantly enriched in these overlap regions. We looked to see what these genes were, and we found nothing that was significantly enriched, and we found no genes related to meiosis, chromatin condensation, or DNA repair. Additionally, we just wanted to double check what's going on with specific key meiotic genes. And we looked at their expression per genome. So here you can see SPO11, RAD51, and ZIP1. And the expression level is stable, indicating that the disrupted patterns of double-strand break and synapsis that we saw earlier in prophase 1 is not due to the misregulation of these genes. So in answer to my second question, I find that no, gene expression in meiosis is not significantly altered due to polyploidization. There's tons more to dig into with those data, but that's what I'll share for today. So next, I wanted to understand if the physical environment of the meiotic nucleus is changing. It's really important to remember that the meiotic nucleus is a dynamic place. Throughout meiosis, there's a purposeful reorganization of the nucleus. You see the chromosomes condense and expand. There's the nucleolar movements, there's chromosome rotations and oscillations. And these are time-lapse videos of live diploid myocytes. These movements are driven by the nuclear envelope. And these dynamics actually do facilitate meiotic mechanisms like pairing and recombination. They promote homologous interactions and they disrupt non-homologous interactions. Previous work in diploid maize shows that the nucleus increases and then decreases in size over prophase 1. And nuclear volume was also shown to correlate with chromosome volume early in prophase. As I was imaging the polyploid myocytes, I observed that this pattern seemed to be disturbed. So to make a long story short, over the course of my imaging, I noticed that the polyploid myocytes were not twice the size of the diploid myocytes. And here on the y-axis, if we look at the nuclear volume per haploid genome. So here we have all of the 6 lines and they're grouped by genotype. And the lighter line is the diploid data and the darker line is the tetraploid data. And in all 3 groups, you can see that in at least 3 out of the 4 substages, the diploid is significantly larger than the tetraploid in terms of volume per haploid genome. So in these tetraploid lines, you have double the amount of DNA in a nucleus that is not double in size. So because I was surprised by this fact, I decided to measure the chromatin volume. So if you think about the meiotic nucleus as a bowl with spaghetti in it, and now we have a situation where we have twice the amount of spaghetti, but the bowl is not twice as big. But it could be that you have like whole wheat spaghetti or something, and the spaghetti is more dense, so it doesn't need to take up as much space. So to see if the polyploid chromosomes are more compacted, I looked at, again, the 3D myocytes, and I measured the volume of the chromatin. So here, again, I'm comparing chromatin volume per haploid genome, and I see that in 2 of the tetraploid lines, there's no significant difference. And in the B73 tetraploid, we find a larger volume of chromatin per haploid genome. So that we can conclude that our spaghetti or chromosomes are not compacted more densely in the neotetraploid lines. And in one line, in fact, maybe less so. So this, taken together with the smaller nuclear volumes in the tetraploids, indicates that the meiotic nucleus in these lines is more crowded, and this can potentially impact chromosome movements and thus recombination mechanisms. So the question remains, why do we see these altered patterns of double-strand break formation in the neotetraploid lines? And why do these breaks persist into pachytene? We find that the tetraploid meiotic nuclei aren't double in volume compared to the diploid meiotic nuclei. And additionally, the chromatin volume does double. So this could have a potential impact on chromosome movements. And if the tetraploid nuclei are more crowded, perhaps it's more difficult for these non-homologous interactions to be disrupted. Another question is why the initial patterns of double-strand break formation in the different neotetraploid lines are different so early in prophase 1. And the answer to this question is going to require future research. But DNA methylation patterns and nucleosome occupancy are known to impact where recombination occurs. In fact, a postdoc in our lab, Mingwei Wang, is doing a lot of really cool machine learning modeling, and he finds that with only information about 2 types of DNA methylation and information about nucleosome occupancy, we can predict crossover sites reliably. And DNA methylation is known to be altered in neopolyploids and could be having an impact in these lines. So I've collected whole genome bisulfite sequencing data of these diploid and neotetraploid zygotene anthers, and I'm looking at their DNA methylation patterns. So the research continues, and I hope to have an update to share with you at some point in the future. So although more explanation is necessary, we can see that the nuclear and chromatin environments are altered in these neotetraploids. And because these environments are so crucial to recombination mechanisms, these alterations are having an impact. Double-strand breaks increase and persist, and these problems from early prophase 1 continue later into prophase 1 to impact recombination resolution. And these data really emphasize the importance of not just studying meiotic outcomes of polyploids, but looking at the dynamics of recombination and pairing earlier during meiosis. I took on a huge project, and I only scratched the surface of the impact that polyploidization has at the molecular level in meiosis. And this system has so many avenues to investigate with amazing germplasm, great cytogenetic resources. So I can only hope that future graduate students and researchers will continue to explore this. And with that, I would like to thank Wojtek and all the people in my lab, my committee, Jeff Doyle, another great polyploidy biologist, and Paula Cohen, Susan McCooch, a bunch of collaborators, and my funding sources. And yeah, thanks. I'm happy to take any questions.
All right. Well, thank you, Mischa. That was awesome. I mean, I really want some of those 3D models for backgrounds now. You know, those are amazing.
Yeah.
I'll as always, we'll let folks pop up their hands virtually or put their questions in the chat box and ask away. All right, Jeanette's up first.
Hi, Mischa. Super cool stuff. I
Thanks.
was wondering whether or not you see any association between the inter-individual variability in pollen viability and the other traits.
Yeah. So I didn't have like so many graphs and so little talk time, but yes, so I actually separated out all of the data of double strand breaks and everything by plant, and it does not vary by plant. So I found very consistent the pattern of the homozygous line being about 1.5x the diploid and the heterozygous being 2x the diploid, even in a plant that, well, so it's hard because I can't track each plant, because to get the material to do the experiment, I have to kill the plant. So I don't know what the ultimate pollen viability of that plant would be, but I imagine if there was some variability in the amount of plants I looked at, I would have seen it.
So then what do you speculate is behind the interindividual variability in pollen viability? That's really hard to say. Do you think it's just like other segregating whatever individual loci?
Yeah, I mean, there could be so many things. I think for sure it could be things to do with recombination resolution. So the double strand breaks are more like the initiation of the whole mechanism. But even in the heterozygous lines, I'm still seeing multivalence. I'm still seeing like lagging chromosomes, chromosome bridges. So there's still lots of problems with recombination resolution that I think are impacting viability.
Cool, thanks.
All right, our next question is from Zishan Sham. If you would like to pop on your audio and ask, or we can just read it from here as well.
Okay, yeah, I can just read it. So if pH1 was responsible for maintaining crossover under control in wheat, then what could be possible for the reason in neopolyploid maize? Yeah, that's really a great question. The pH1 locus in wheat has been studied a lot, but as far as we know, it's pretty unique to the wheat system. I mean, there are other loci that do similar things in like Brassica, there's PRBN, but that functions, it's a totally different locus. It functions in a totally different way. So it seems like these stabilization mechanisms evolve sort of in each lineage, and they're, at least as far as we can tell so far, there's not really a consistent theme. So I don't think there is a mechanism in neopolyploid maize, would be the short answer to your question.
All right, Brittany.
So first off, I would like to just compliment you for like the clearest explanation of how crossing over works that I've heard.
Oh, thanks.
It was really good, and I think I understand it now.
Oh, thanks. Yeah,
So,
I think I went to so many meiosis seminars and stuff, and I really think that our field should like link up a bit more. But thank you for the compliment.
So my question, and this might be kind of a naive one. So when these, single strand breaks are sort of looking for their partner, how identical do those stretches have to be that they're looking to hook up with, and how long a region are they looking for?
Yeah, okay, that's a great question, and like a super active argument in the meiosis field. So we actually know that these resections are pretty long. They're like 500 base pairs to about a kb. Okay. So we're talking about like some pretty, it's not just like a little 20 base pair primer. And as to how similar they have to be, it seems like it varies depending on the species. So they've done a lot of work on this in mouse, but in plants, we don't really know. And one of the sort of crazy ideas that I had at one point that hopefully I or someone else will be able to do is we have all these different inbred lines of maize that are sort of at different degrees of sequence divergence from each other. If we could make like a series of polyploids, or even just hybrids to get at the question without dealing with polyploidy for a second, then I think we could get at that question. But we do know that there is a mechanism to discourage repair by the sister chromatid. So that would be the identical strand. And so that's discouraged. And so it wants to promote homologous rather than sister. But how that mechanism exactly works, we don't know yet.
I think if y'all could answer that question, that would be like the holy grail of the question of, auto versus allo.
Yeah, I think, as in a lot of things in biology, it's going to be different in different species, at least. So looking at this sort of level of meiosis, there aren't a lot of species that have been investigated. It's basically like Arabidopsis, maize, mouse, yeast, C. elegans. And there are some definite differences between those, but yeah, I agree. It's a fascinating question.
Yeah, thanks.
All right. Our next question is from Justin Conover.
Hey, Mischa, thanks for a great talk today. I really enjoyed it. Early in your talk, you described kind of the three outcomes of the double-stranded break. I'm wondering if you can classify how many of those three classes occur versus homologous versus homeologous pairing, and whether you see a difference in those.
Yeah, that's a great question, and something that sadly COVID prevented me from answering in my dissertation. We actually have antibody markers in the lab, or we made them, against a protein that marks type 1 crossovers, which are the ones subject to interference, and one that marks class 2 crossovers. So those like two separate proteins. And I was hoping to do similar immunolocalizations to understand that. Like, one hypothesis is that the class 1 is sort of the predominant. So like in diploids, 90% of crossovers are class 1. And then class 2 is like the remaining kind of 5 to 10%. So that those numbers vary in different species. But the idea is this class 2 pathway is kind of the backup pathway. Like if an intermediate gets messed up or is kind of weird or is between like two really divergent sequences, then it kind of gets shunted into this class 2 pathway. So one of my hypotheses is that we have an increase in crossovers, but in this class 2 pathway. But I wasn't able to do those experiments because the lab shut down and stuff. But hopefully someone else will take them up, or wherever I go in the future, I'll be able to do that.
Cool. Yeah, hopefully somebody does. Can you say a little bit more about the non-crossover pathway? Like what percentage falls into that, and how would you detect it?
Yeah. So gene conversions. So in just normal diploid maize meiosis, there's about 500 double-strand breaks, but 20 crossovers. So there's 480 gene conversions. Detecting them is really difficult because they're pretty short. They're 500 to 1,000 base pairs, and they, if there's not a SNP there, or if there's not a good marker, they're really hard to detect. So there's actually a lot of groups working on that now, on sort of bioinformatic pipelines of how to detect them, where they happen, and that kind of stuff. I think it's an ongoing question, but yeah, the bioinformatics side's not my area of expertise, but I know there's a lot of work going on there.
Thank you so much. I really appreciate it.
Yeah.
Let's see, one last question there from Zishan Sham again. Do you see any zip working in polyploids?
Okay, so we didn't look specifically to see if there was like a different variant of the ZIP1 gene. I mean, we have the mRNA from the RNA-seq that we did, and we did some aligning and stuff, and we didn't see any difference from the diploid. So I don't think it's a different ZIP1 variant in the neopolyploids.
All right, very cool. I have a real quick question. So you may not be able to tell this. I've always been fascinated sort of by trying to figure out the, and reading about these the origins of replication and stuff like this. What are they? The nature of those in plants, and sort of looking at your work, do you see evidence that there at least where these double-strand breaks are occurring, or where the Rad51 localized to was always in the same areas, or does it look like it just moves around? What's your sense of? Yeah.
That's a great question. So we haven't done any of this work in polyploids because bioinformatically that's, you know, a whole other ball of wax, as you all know. But in diploid maize, one thing that our lab is starting to do is a lot of ChIP-seq with these antibodies. So we've done ChIP-seq with the RAD51 antibody that I showed. And really interestingly, we find that there are hot spots of where double-strand breaks are formed, but they're very uniformly distributed across the genome and along the chromosome, which is really interesting because the maize genome is full of these repetitive elements. A lot of the genes tend to be, you know, towards the chromosome ends and not in the pericentromeric regions, but we find double-strand breaks all along the chromosome. And that kind of goes, so, you know, if you're using this as a homology search process, you kind of want to be able to align all along the chromosome. But crossovers predominantly happen at the ends of the chromosomes. There's this U shape. So kind of reconciling that of like, okay, we have breaks everywhere, but we only want crossovers here. What pushes those breaks to form crossovers? Yeah, seems to be like chromatin state, DNA methylation. There's some evidence that there's like actual sequence specific cues, but it's an ongoing question.
That's fascinating. I love it. So I want to connect. I might connect with you later about some of this other work that you mentioned with the machine learning. That the other person in the lab's working on too. I think that's so fascinating.
Yeah, he's getting ready to submit a paper that I think is on that and on his models and stuff. And I think it's super fascinating.
Yeah. Very cool. Well, I think that's all of the questions that are posted here. I know that Carl had a question from before, or has it not been? Carl's waving me off. I could see in the box. So, all right. Are there any other questions for either Brittany or Mischa? Loud construction. Oh, okay. All right. Well, I really enjoyed both of these talks. It's so awesome to see what both of you have done. And I know Mischa applied to come to my lab back a long time ago, and I'm happy that she didn't because she's done great, amazingly cool things that has brought some new things into the polyploidy community that she certainly wouldn't have been able to do here. It's just wonderful to see this great work. And I appreciate everybody for participating in these talks. There are now, I believe, 24 hours of talks on polyploidy posted up on the website and through the YouTube channel there. So this is all possible because everybody is signed up, and I appreciate everyone continuing to do that. We'll take a little bit of a break for the summer here, and we will be back in the fall. I'll email folks over the summer. I might tap a few other folks to help me organize and coordinate here for the next year as I get back to full scale teaching here in the next academic year. But this has been so awesome, and I want to keep this going. I see no reason why we can't have this. I originally envisioned this to be a big lab meeting during the pandemic. I didn't really know what it was going to be like. We usually have about 40 to 60 people on live, and then just so everyone knows, the recordings get watched another 100 to 200 times typically. And there are about 700 people now signed up for the mailing list. It's been impressive, and I thought we'd have like 30 people I know that would just join us on the mailing list, and maybe 10 people would show up to the talks. So this has been really fun. And I want to keep doing this with everybody. It's just so enjoyable to see everyone and hear about all this exciting research. I mean, it's so fun, and I appreciate everyone taking the time to put talks together. That's really the hard work, and so all of you make it happen. All right. Well, I'll see everybody in the fall. We'll see everybody at the botany meetings this summer, evolution meetings. There's SMB. Well, we'll see everyone online at those meetings. Hopefully everything will be returning to normal here again in the fall. I think vaccinations are rolling out in many more places, and keep those going through the next year. And hopefully we'll be able to see everyone again in person in the not too distant future, and get to hear about everyone's research. But in the meantime, everyone have a great summer. I'm going to go out and I've got my presses ready. I'll be out collecting in Colorado this weekend, so that's my big plan for vacations. Hopefully, everybody can get some fresh air and get out and botanize. All right, we'll see everybody. Bye. Yes, Carl, I'll be looking for the ferns.